Hello everyone, my name is Ruth Bamiboye. I've got Priyanka Sharma here with me. Today, we will be presenting the research paper, Global Analysis of Protein Expression of the Inner Ear Hair Cells by N and his colleagues, published in the Journal of Neuroscience in the year 2017. The inner hair is significant in maintaining hearing and balance. It consists of the sensory epithelium, made up of the cochlear and vestibule. The cochlear is also known as the auditory epithelium, which changes sound waves into nerve signal. The vestibule sensory epithelium found in between the cochlear and the three semicircular canals assists in balance and posture to achieve equilibrium. Hair cells are flag-shaped ciliated cells found in the sensory epithelium. It plays a key role in amplification and synaptic functions via highly specialized hair cells proteins within the inner hair. Genetic disorder, infectious diseases, high level of noise exposure and high dosage of octotoxic drugs can cause damages to the protein machinery of hair cells, including stereocilia transduction apparatus, presynaptic signaling complexes, and intracellular mechanism for supporting sound. Although 5% of the world population has disabling hearing loss, World Health Organization estimated an increase to over 900 million in the year 2050. This figure 1 is a structure of the inner hair cell. This research paper aimed to establish the inner hair proteome using shotgun proteomics. Materials and method. Transgenic mice with POU4F3 genes involved in development of the middle and inner ear was used. This gene was validated in the internal hair by immunolabeling and qRT-PCR for expression within the hair cells. Wild mice was also used. Samples collected include internal hair, the whole internal hair, sensory epithelia, and the hair cells. Sample was isolated by microdissection and enzymatic dissociation. For high purity, HC samples were isolated by FACS, which was then precipitated out using methanol and chlorophyll. For LC-MS, sample was first digested with trypsin overnight, followed by fractionation. The fractionated sample was directly electrosprayed into the spectrometer. Peptide spectra was analyzed for protein identification, quantitation, and mapped out to gene responsible for deafness within the hair loci. The results are in discussion. Figure 2A shows the inner hair proteins before sorting out and the GFP positive HCs after sorting out. Figure 2B shows the confirmation of the GFT positive hair cells with the dimyo7A. Of the 12,712 total proteins identified, half is found in the purified HC and other cells, of which 934 is specifically expressed in HC. Among the proteins expressed in the specific to HC, some proteins support auditory and balancing. This is shown in figure 3A. Gene oncology related to ciliated cells of HC support the possibility that HC-only proteins likely represent both HC-specific proteins and some low-abundant proteins of other cells. This is shown in figure 3B. Rank abundance was measured semi-quantitatively using NSAF divided by two sets of data, HC-only proteins, those specific to HC, and HC-enriched proteins, those with significantly high abundance in HC samples. Figure 3D and 3A confirm these proteins to be 92.
The next step was the mass spectrometry analysis of HC proteins, which yields novel and isoform-specific HC gene products. The approximately 12,000 identified inner ear proteins represented thousands of genes, including hundreds of genes with products identified only in the HC sample. The 934 HC-only proteins mapped to 458 genes. However, only 351 of these genes are considered HC only, whereas the other genes have at least one protein form identified in a different sample. The resulting gene products were identified by the MS approach and compared to recent cochlear and utricular RNA sequencing analysis performed by Scheffer and his colleagues. Scheffer purified GFP-positive HCs from the same transgenic mouse line as the current study and examined gene expression in pooled HCs versus pooled GFP-negative supporting cells illustrated in Figure 3a. For comparison, the authors mapped the proteomic data to MGI gene identifiers and used only transcriptome data from matched ages, shown in Figure 3b. Scheffer and his colleagues examined differential gene product expression between GFP positive and GFP negative cell populations, whereas for this study they performed progressive enrichment for GFP positive cells by comparing to IE, SE, and HC samples. They analyzed the RNA sequence data and found that 2.9% of the genes were found exclusively in the GFP positive dataset, as shown here in Figure 3c, the top part. And at the bottom here, the MS result showed that 40% of the identified genes were found only in the GFP positive dataset. As the overlap of the genes in the MS-HC only and GFP negative datasets is minimal, the HC only gene population is relatively unchanged. 89.2% of the HC only genes identified by the enrichment strategy are also identified as HC only by the GFP positive versus GFP negative comparison. Importantly, MS analysis of GFP negative cell extracts failed to identify any expressed GFP peptides, which shows that these cell extracts have very little or no HC contamination. The FACS purification of HCs yielded enrichment of HC gene products that was in many ways similar to previous mRNA studies. However, when comparing the HC gene products identified by both approaches, HC gene products identified only by RNA sequence tends to have a lower abundance, which is shown in Figure 4d. The next step was to select several proteins for validation of HC-specific expression by immunolabeling based on criteria such as novelty to specific tissue type, to inner ear and standing for deafness genes. Several peptide sequences for PARP3 were identified as HC only. The authors validated vestibular expression of PARP3 via immunolabeling of utricular hole mounts and observed highly specific expression in the HC cuticular plate region, which was consistent with the previously reported role for PUCs in the development of cochlea. Another example was of neurolegin 3, a glutamatergic synaptic adhesion protein, which was also identified as a HC-only protein. When it was mutated, it resulted in impaired auditory or vestibular phenotypes in mice. Moving ahead, the mutation-driven auditory or vestibular impairments. Hearing loss or vestibular impairment can arise from damage or mutations across different inner ear cell types. However, the authors hypothesized that the changes in HCs are likely to produce a measurable phenotype. To test this possibility, they compiled phenotype-genotype associations from the mouse genome database from transgenic mouse assays related to inner ear structure and function and examined the distribution of these genes across their datasets. Genes related to transgenic mouse hearing or balance behavioral deficits such as ESPN and GRXCR1 were overrepresented by gene products in the HC-only sample compared to IE or SE-only samples as shown in Figure 6a. In contrast, genes related to impaired eye physiology, which was used as a negative control, were not statistically overrepresented in the HC-only group. Overall, proteins associated with impaired hearing phenotypes are relatively more likely to derive from HCs. Now, more directly related to human health, the distribution of known human deafness genes was identified by MS. Deafness gene products found were overrepresented in the HC-only group compared to SE and IE group. Currently, only a small percentage of the causes of auditory and vestibular hearing loss can be treated with hearing aids and cochlear implants. This research paper has taken a step in by compiling the most complete mammalian inner ear protein expression catalog to date. With a prediction and increase in hearing loss by WHO, further research is critical for thorough understanding of HC gene expression at the proteomic level to clarify normal and aberrant HC structure and function, sources of dysfunction in hereditary deafness, and developing potential therapeutic treatments. Researchers compiled the most complete mammalian inner ear protein expression catalog to date with the identification of HC gene products not previously identified. However, the description of the inner ear proteome remains incomplete and many more important proteins are still unidentified. This can be due to several factors such as very low abundance of many proteins, potentially poor extraction of multipass transmembrane proteins, proteins with amino acid sequences lacking the appropriately sized tryptic peptide fragments, the absence of protein amino acid sequences in the reference protein database, alteration of proteins by post-translation modification, and less sensitivity of proteomic MS than RNA sequence analysis. Future research should aim at mitigating these effects by combining both MS and RNA sequence analysis for comprehensive gene and protein expression of all inner ear cell types. Thank you.